I've just arrived outside the blizzard, where I'm going to interview two scientists funded by the Dr. Hadwin Trust on their work. So, could you introduce yourself?
Yes, I'm Professor Mike Philpott, so I'm Professor of Cutaneous Biology at Barnsley London Medical School.
Right, so what do you do here?
I carry out research on skin. My main focus nowadays is on skin cancer and also trying to understand what controls hair growth.
So, can you give us an example of a technique you're using?
Yes, probably one of the techniques we use here is we do a lot of cell culture. So we take cells out of human skin and we grow them in a dish in the laboratory, and then we can actually put them together so they actually make a whole skin in the laboratory. And often we then put into those skin cells genes that we think may cause cancer and see whether we can actually develop tumours actually in the dish. So we're very interested in trying to understand that disease, and we're also very keen in trying to understand psoriasis and trying to treat that as well.
Right, so what can we do to help the Dr. Hadwin Trust?
I think with the Dr. Hadwin Trust, what we are trying to do is get everybody out there who works on skin to realise that we don't really have to use animals to do our research, that there are a lot of very good cell models. We can grow human skin, we can grow human skin in the laboratory and make a very good epidermis, and so a lot of our research that we can actually do, we can do in human cells and human tissues. I would argue that animal skin, and especially mouse skin, is very, very different to human skin. And so that mouse skin is a very, very poor model for human skin. Mouse skin is only 2 or 3 cell layers thick. Human skin is about 30 layers thick. So the biology of human skin is very different to mouse skin. I think my ideal world is a world where we basically have human models for human disease. And so I would like to see much more funding for research being put into research that's using human cells and human tissues.
Could you introduce yourself?
Yes, so I'm Dr. Adrian Biddle. I work here at the Blizzard Institute as part of the Dr. Hadwin Trust's Centre of Excellence in Animal Replacement Research with Mike Philpott.
Okay, so some might question why we shouldn't test on animals when it's simpler. So what would be your answer?
Well, the basic answer is it's not simpler. People are moving towards these new in vitro models, models in a dish, because they actually they've been shown to be relevant to human biology, but also they're simpler and what's called more tractable, which means that you can do the experiments more accurately in them and get more information, as opposed to an animal model. As you can imagine, you get a set of mice, for example. Each mouse is going to be different.
So did you always want to be a scientist?
Yes, yeah. Actually, since I can remember when I was about 7 years old, thinking I wanted getting the science encyclopedia that my school teacher gave me, and from then on, really, I've never even considered anything else.
So, if you're a young scientist, we hope you learned something today. That species-specific research can be much more accurate than animal research. This is Freddie Jemison from Trinity School, reporting for BBC School News Report.